Thank you, Nick, for the nice introduction. I hope you can hear me well. So I have confession to make. James did not have just random injury. I felt jealous that he was supposed to give this talk, not me. So when he was sitting, I just came from behind, twisted his neck a little bit. Now he has pinched nerve. He couldn't come. I'm giving the talk, so mission accomplished. So yeah, I wish him speedy recovery, though. And it's an honest wish. So I'd like to show you at the beginning of this talk a movie which, in some sense, is, for many of us, is a dream. So this is a movie of it's a development of the bee, so from larval stage to almost adult organisms. And I think it's true that each of us somehow wishes that we were able to generate this movie using our simulation engines. We're not there yet. We have bits and pieces. We developed tools, we developed models, we developed models which address parts of the development, but we are not able yet to integrate all those models into a master model that would actually be able to generate a simulation which would allow us to study all sorts of different sorts of perturbation that can occur in the development of insect in that case. And if we can model insects, we can model pretty much everything, right? Because cells, regardless where they are, they always redeploy the same set of behaviors. And they have, they can grow, adhere to each other, move spontaneously, secrete signals, absorb signals, and die. And all those behaviors effectively feed into the development of either organism development or disease development or disease progression. So understanding how the cell's behaviors function and how cells function when they interact with each other, it's essential for building predictive models in biomedicine. So I think it's fair to say that all of us, or large part of us here, are tissue modelers. Okay? So what we are building is effectively virtual tissue infrastructure. So what is this virtual tissue? So virtual tissue, to me, it's a workflow which begins with experiment, progresses, and then goes to formulation of qualitative biological model. Then we build quantitative model, which usually is a collection of different submodels. So we can have whole body scales via PBPK modeling. We can have cell-based modeling, and this workshop addresses this part. We can have subcellular modeling. And then we build integrated model. This model is then being put into simulation engine. The simulation engine runs, generates results. The results are being analyzed, and then the results are being compared to experimental data. And then this experimental data, the experimental data is used to refine model, and the overarching goal of virtual tissues is to generate and encapsulate knowledge. Okay, so that's what we are doing here. So this is a simple cartoon which probably summarizes the mathematical complexity of multicell modeling very well. So we have basketball player, okay, and we can easily build a model which takes as an input parameters the velocity vector, various distances, and as an output, it calculates trajectory of the basketball as a function of time. Okay, and this model can be used to predict whether the shot was successful or not. Right. So why am I showing you this? Because multicell models are effectively simpler version of that cartoon. Instead of calculating trajectory of the basketball, we are calculating trajectory of each individual cells. Some of us will have 1,000 cells, some of us will have million cells, some of us will have billion cells, or even more, depending on the problem. But the goal is always the same: calculate trajectory of the single cell. Why is it simpler? Well, it's simpler because we assume that force is proportional to velocity, not acceleration, and that makes your life much easier. So why do we bother? Why do we even try to calculate those trajectories of the cells? Well, it happens so that many or most of the tissue diseases happen because cells are in the wrong place at the wrong time. We can look at cancer, we can look at polycystic kidney disease, we can look at age-related macular degeneration, we can use that at various granulomas, which are tumor-like growths, but not invasive. And therefore, calculating trajectory of cells makes a lot of sense. Of course, because cells are the building blocks of tissues, right? It's they are not the only ones. There's extracellular matrix, which plays also a profound role. But because cells are building blocks of tissues, again, calculating trajectories makes sense. Models which we build, mechanistic models of multicell models of tissues, they can be capable of explaining how gene mutations in few cells results in a tissue deformation. Okay? I'm not going to give you some specific examples, but we are publishing a model on polycystic kidney disease, and it happens so that single mutation in a single cell can lead to cyst formation, to multiple cyst formation, which renders kidney dysfunctional in the long run. Okay? By building models, we can do controlled virtual experiments. Okay? When we want to perturb biological system, we don't perturb just one aspect of the pathway. Usually there's a crosstalk between pathways. When we are on a computer, we have the luxury that we can actually perturb exactly what we want to perturb and study the effect of this perturbation on a system. And in the long run, we hope that those multicell models will replace experiments. We're not there yet. Many of us work with experimentalists, with clinicians, and I think slowly we are getting to the point where there is a better appreciation of what we are doing, but there's still a long way ahead of us. And which models are worth building? Mainly those models which help biomedical researchers understand biological mechanisms. Okay, so here we are. So let me show you slightly an example of a model that we've built 6 years ago. It's a tumor growth model. So we start with pre-existing vasculature, and then we have tumor cells which grow in response to nutrient availability. The cells which are in the middle of the tumor, they are deprived of nutrients. They secrete VEGF signal. VEGF signal stimulates vasculature growth. Vasculature invades tumor. And then we can study 2 scenarios. What's going to happen if we allow vasculature to grow, and what's going to happen if we suppress. So this is just an example. And keep in mind that this model, what this model does, it actually calculates trajectory of single cell, of each individual cell. Okay? So these purple segments are neovessels, which are secreted in response to hypoxia-inducible factor. This HIF factor is secreted by starving cells in the middle of the tumor. So those neovessels, they invade the tumor. Okay? Now, if we run the model, another model, we can ask ourselves, what's going to happen if we suppress the formation of those vessels? And you will see that in that case, tumor becomes more migratory. So the tumor starts in the middle, but as time progresses, the tumor moves from the middle of the lattice to the side of the lattice and aligns itself along the blood vessels. Okay? So this is just an example of models. I'm not going to talk too much about modeling. This talk is not about modeling. This is just to show you that, indeed, all we are doing is just calculating trajectories of cells. And it's also good to look at this slide, which shows modeling scales in biology. Okay? So when we're talking about multicell modeling, we are probably addressing those 3 scales: organ scale, tissue scale, multicellular scale. Again, whole animal is a dream. Okay? Keep in mind, 1 cubic centimeter of tissue, that's 10 to the 9 cells, roughly. Okay? So it's a dream, but, well, will it be achievable? Who knows? But those 3 scales, organ, tissue, and multicellular scales, are certainly achievable. Now, we can put our multicell models in the context of the whole animal model. We can link multicell models with PBPK, with models like PBPK, which operate at the whole animal scale, and models which operate at the intercellular, cell, and subcellular scale. So this is all doable. And then the techniques are listed here. So they range from, of course, ordinary differential equations, fluid dynamics, PDEs, Agent-based models are the domain of multicell models. Continuum mechanics also apply to the tissue scale. Okay? So that's where we are in terms of science and biological scales. So how do we simulate our, how do we run our multicell models? They all pretty much follow the same principle. So first of all, we assume that force is proportional to velocity. Okay? Cells live in viscous medium. We can neglect acceleration. Okay? Next, for each individual cell, we compute forces or energies that drive self-motion. Okay? We use auxiliary equations such as reaction diffusion, Navier-Stokes, that capture the interaction of cells with environment. Okay? We compute trajectory of the cell, and we go back to the point 1. Okay? And usually, when we deal with forces, we have to deal with a system of first-order ordinary differential equations, which, as I said, makes your life much easier. So I'm going to show you several of the possible approaches to building cell models. So probably the simplest one, multi-cell approach, is cell automaton, where each cell is represented as a point in space on a lattice. Okay? So we have models from Sandy Anderson. We have models by Holger Perfal, Oxford group, and Nottingham group, vascularized tumor. We can build generalized agent-based models. So we have models by Sasha Popel, arteriosclerotic model by Shane Pierce Cutler, and model by cellular aggregate by Stefan and Dirk. So those models, they represent cells as a single, as a point in space. So we can be either on lattice or off lattice. Another class of models is the class of models where allows you to represent cell shape explicitly. So we have, for example, red blood cells models, which come from a group of George Karniadakis in the Brown. So he models, those are very nice models, very powerful, also very time-consuming. So he models the deformations in red blood cells. We have Potts model. I showed you the movie. We have subcellular element model by Tim Newman. Immersed boundary method. Kasia Reiniak from Moffitt is one of the experts in this domain, vertex models. And the nice thing about all those models, I mean, the nice thing, ideally, in the ideal world, all of those models for a given biological model should give you the same answer. So if we have biological simulation, no matter which model we use, we should get same, at least, qualitative answer. Now, there are many software implementations for those models, okay? So first let me start with center models, because it turns out those models have probably most software implementations available. So we have PhysiCell, Paul Macklin. We have EpiSim simulation, which comes from Heidelberg. Biocellion, new kid on the block. Cellsys, I think this name has changed. Stefan, I don't know what it is right now. You have to talk to Dirk and tell me what the name is. But anyway, it's a code by Dirk Drasdo. And we have, of course, Chaste. And Chaste is not just a center model. They have multiple implementations of various models, but center model is definitely one of their strong domains. So when we go to software implementations that allow, yes? Yes? Oh, I'm not going to tell you because I will get the star of shame. That's a nice trick question.
Some of these will be given demos later.
Yeah. Yeah, well, I'll actually, I have a slide. I will briefly talk. I'm not going to say anything bad about them, I promise.
We have some discussion time.
Yes. So actually, I usually give a talk where I actually talk about strengths, I mean, pros and cons of various platforms, but I won't have time because I'd like to go to other issues that I think are more important for this community. Okay, and I think that I mean, the whole goal for this talk is to come up with some talking points for this conference. And I think there are issues which go far beyond the details of the implementation of the models. And I will talk about them. You will like that, I promise. So here we are, software implementations for methods which allow explicit cell shape representation. So cellular pods model has 1 to 3 representations. CompuCell, Morpheus from Dresden. There is a Simune code which comes from NIH. And there is a tissue simulation environment by Roland Merckx, which I don't think it's that. I mean, most of the code of his code has been included in CompuCell 3D. There's, of course, Virtual Leaf if you want to study plants. So Roland Merckx has Virtual Leaf, very nice platform. Of course, there is a code, LAMPS, which is the code developed by Sandia National Lab, and this is molecular dynamics code, which has been repurposed by Karniadakis and colleagues to study red blood cells. Okay, and of course, perhaps there is more. I might have skipped something, so please don't get offended if I did not include your platform. So now the slide with software. So again, this might not be the complete list, but we have Physicell for center model from Paul Maclin's group, Morpheus for cellular pods model. It's similar to CompuCell 3D. EpiSim from Heidelberg, so it's Niels Grabe and Thomas. Biocellion, front row, you guys. We have Simune. Well, CompuCell 3D I mentioned already, so there is no point. Chased. So Chased is, among all those, is probably the only platform which is multi-method. So they have multiple implementations, okay? So they have pods model, they have vertex model, they have center model, they have cellular automaton model. So, and I'm sure you will hear more about Chase from either Helen or Alex, so if I let them talk, of course. And we have Virtual Leaf for plant modeling and CellSys software for center model, which comes from Dirk Dresde group. Did I say anything funny? Well, ask him what chaste means, really. So I will tell you what chaste is. It's Chairs Against Sex Trafficking in Europe. That's what it means. That's what I found. So seriously, it is. If you type chaste in Google, that's what comes up. So some of those tools are closed source, some of those are open source. So now here is, I was browsing Slashdot in the morning, and this is what I found. So this is an interview with a guy who was running a company for 4 years, and then all of a sudden, recently, yesterday maybe, he decided, okay, we're switching to open source. Why did he say that we are switching to open source? Well, if you read this, his answer, he said being proprietary was a barrier to adoption. There will be other consequences of that, and I will talk to you about the social dynamics of being closed source and versus open source. But I can tell you that if you're afraid that by going open source, somebody else will use your code, I think your thinking is wrong. I think you should be afraid that nobody else will use your code, because this is the problem right now, and we'll talk about it soon. So that's of course. There is a period of the development in which you are better off hiding your code if you want to get ahead of competition. But if you're doing this for 10 years, then probably you should change your mindset a little bit. So I'm not going to talk about those. So let's talk about how do we construct our simulations. What are the simulation components? So we deal with objects, and objects can be their cells, can be fields, can be extracellular matrix. I think that's more or less the summary. And objects have behaviors, okay? So cells can have a volume, they have properties, they have volume, surface, the Young modulus. The fields can have diffusing constant, they can interact with other fields. And of course, we have initial conditions, the data, which is used to parameterize models, okay? And data does not only parameterize initial conditions, it also parameterizes how cells behave, okay? And then we have, of course, processes. So this is how we represent cell dynamics, which type of equations we use to describe chemical dynamics and so on. Okay, so those are the components of the simulation. And typical simulation workflow looks like that. We start with biological observation, then we choose methodologies, and we implement the simulation. Okay, And this is fantastic. This is quite the quick and dirty method. The problem with this workflow is that it's not reproducible. It's called hacking, so to speak. A better workflow is more involving, and it's more systematic, and I'm going to walk you through the steps, but at least gives you a hope that your model in the future will be reproducible. So we start with biological observations, okay? And then we define initial hypothesis and define qualitative verbal model. Okay, so we are saying, what are we modeling, what are we trying to check, and what are the components, what the cells do, how do they behave, biologically, no equations whatsoever. Okay? After we did this, we have to ask ourselves, how are we going to model this model? How are we going to build it? What are the subcomponents? Are we going to go to subcellular scales? Are we going to connect to whole animal model? Okay, so that's the next step. Based on biological information, we have to create initial conditions. Okay, so usually, I mean, if we are lucky, if we work with clinicians, it can be MRI scan, it can be CT scan, or it can be microscopy data. Okay, and we need tools for that. So for each of those arrows, we need tools. Okay? Now, when we put those slides together, so we have qualitative model, which then we turn into quantitative models. Okay? We combine it with initial conditions, and we build a template, a model template, for that integrates subcellular, multicell level scales and perhaps tissue levels. Why is it a template? Because it doesn't have parameters yet. Okay, so there will be parameters which will feed in. And this is precisely where this step is talking about. So we take this model template, and then we look for parameters. And again, where do we look? We can either talk experimentalists, we can scan databases, but to be systematic, we need to also develop, at some point, tools which will facilitate either mining or retrieval of existing parameters or parameter scans. Okay? And this brings us to instantiated model. Then we, of course, we have simulation engine, and most of us so far concentrated on this aspect. We were building simulation engine, somehow neglecting the other infrastructure. And that's natural, because engine is the most important part. Just like engine is the most essential part of the car, but engine by itself is not a car. Okay? So if you want to build infrastructure, yes, we need to have decent engine, but we also have to have wheels and maybe a few other whistles. So now we have simulation output. Once we have simulation output, which comes from our simulation engine, we can compare it to biological observations and either refine our model or generate knowledge at this point. And that's what this slide represents. So in a sense, the whole idea is by gathering simulation output, instantiated model, biological observations, if we put that together, if we can archive it somehow, we'll have entire workflow stored and ready to be reproduced by other teams. Why is it important? It is important because if you want to address clinical aspects of, if you want to address clinical problems or apply our models in a regulatory setting, we should be able to reproduce our models. Nobody will make a regulation that, hey, we should not use chemical A because our model shows it's harmful if this model is not reproducible and verifiable and testable. And this is precisely what the slide says. So far so good, but what would reviewer number 3 say about state-of-the-art simulation? So those are real critiques from one of our grants. And I'm going to read them because they, just as they affect us, they will affect you or us as a community at some point. So I'm not ashamed to show it. Okay, so I was the PI on the grant. This is what they say. Despite commendable efforts to promote ease of use, the complexity of the system seems likely to make it unusable by biomedical domain experts. Well, how simple can it be? Of course, it can be simple. And we can build tools that will make access, that will make multicell modeling much more accessible. But we should not expect that multicell modeling will be as simple as playing Angry Birds. I mean, I cannot go to brain surgeon and say, hey, look, you need to simplify this surgery because I like to do it on the weekend. I don't have time to learn it. But I think I think some of us, and I think James Glazer is one of those people who tours the world and tries to sell this vision that modeling will be done soon by people who are not core modelers. I think we can move in that direction, but we should be also realistic. Okay? So there is a fine line. If you want to build complex models, don't expect that those complex models will be super simple to build. There is a certain amount of expertise that's required to build models, just like there is a certain amount of expertise required to do brain surgery or any other type of surgery. So the user community remains small, with so far limited impact in terms of publications, and those are almost exclusively in the modeling literature. So what they say is that we are small, in a sense. I mean, CompuCell, we've been around for a while. We organize lots of workshops, and we are still considered very small. Yeah. But that's right. That's right. But it's true that our community, and actually, if I go to the next slide, and I will come back to the previous slide, this is archive submission rate statistics. So if we look at different fields, of course, archives are dominated by physicists, okay, or mathematicians. But if you look, there is QBio, and QBio is this little tiny fragment here, okay? So as a community, we are still small. And it's true, it's small. I mean, we are small because how can we be big? I mean, many quantitative biologists will go for network modeling. Network modeling, I mean, it's difficult, but it's significantly simpler than building models where you have to harness billion cells. I mean, there's no denying. I mean, network model runs for, let's say, 10 minutes if it's complex. Multicell model can run for a month. So there are some expectations from the reviewers which might be unrealistic, and maybe they don't realize that. Another criticism: the directions for planned improvement of simulator appear useful, but not greatly technologically innovative. GPUs, MPI, what else can we do? I mean, but I make notes, and because sooner or later, when you write grants, you will be criticized like that exactly. So, what did he say? Software. I'm not gonna criticize because I'll get a star of shame. I'll tell you offline. Plus, this is being recorded, so I cannot really say everything I like to say. So the environments for faster computation seems unlikely to advance state of the art that will have broader impact on the modeling community. Again. And of course, there is a question of business model. How do we run? How do we formulate? How do we form teams? I mean, apparently, it's not okay if somebody commits his time full time to a project, right? It's being frowned upon. It's probably best if I spend month here, month there, because as we know, this is the best. This is how things work, Not really. If Steve Jobs did not commit to Apple, there would be no Apple. If Gates did not commit to Microsoft, there would be no Microsoft. And that's why, Yeah, I'm modest. I'm very modest. Yeah. So those are, hopefully those are the talking points that we might discuss during this workshop. And see where, as a community, we could go. Because I don't think that going solo and applying for those grants here and there, hoping to pull out $1 million or $2 million for this project or that will be, this funding will evaporate in, let's say, 3 years, is the best strategy. I think we need to think ahead, think about the BRAIN initiative, right? I mean, we need more publicity. We need more critical mass to move forward. I don't think that at this point I don't think that focusing, being selfish is the right strategy. Well, but they have valid points. I mean, it's just as, yeah, of course, it was wrong panel, absolutely. But still, the remarks that the community is small shows up all the time. So I think going forward, nothing best summarizes the strategy that we should adopt as this quote: if we don't hang together, we are guaranteed to hang separately. So that quote is attributed to Benjamin Franklin. Apparently, it was first written down by Frederick Reynolds in his play Life. So what should we learn from other communities? So if you look at SBML community, right? They have standard, they have 200 tools. High energy physics, another big community where nobody builds their own physics simulation toolkit. There is something called root, which everybody shares. Okay? So there is, and of course, there is a question of who's going to pay for it at the end. So in high energy physics, it's clear. They have CERN, they have contributing countries, and there is leadership which says, okay, we have to spend part of our budget developing software. Okay? We don't have this yet. The question is, how do we structure collaborations? What would be the collaborations with, between teams? How much do we want to share? How much do we want to overlap with each other? Okay? How much effort do we have to spend talking about ease of use, ease of analysis of modeling results? Who is our target audience, right? It's clearly that looking at many tools, whether it's Multicell or Dirk's code or Paul Mackley's code, there are rooms for improvements in terms of ease of use. Reproducibility and model sharing. Okay, again, this is pretty much non-existent right now, and we have to address this sooner or later. Standards. These issues comes up sooner or later in the discussions. I have to tell you, standards for multi-cell modeling are hard to develop. If you think carefully, the only underlying standard for multi-cell modeling is biology. Okay, because it's very hard to translate cellular automaton model into, say, POTS model or subcellular element model. I mean, those models speak different languages. It's a mess. And we had several meetings. Cell behavior ontology came up out of that, but we don't have good standard for multi-cell modeling. And we have to sort of address. My personal view is that we have to focus first on building next generation easy-to-use tools. But that's my opinion. Sustainability, business model. We talked about in the previous slide, and model validation, cross-validation. That's important if you want to go to clinical setting or be treated seriously by my biomedical community. So I guess let's look at the success story. So I've read the survey, and somebody said, oh, I use, for multi-cell modeling, I forgot what they use, and they said, but MATLAB sucks. I use also MATLAB, but MATLAB sucks. And yeah, maybe MATLAB sucks, but this is really the tool that people use. So yes, SBML community has standard, systems biology markup language, yet I'm just guessing. 60–70% of models which deal with bionetworks are done in MATLAB. Why? Why is it so? Exactly. Fantastic. So this is exactly. So this is absolutely. But there is more than that. So first of all, what MATLAB comes up with is easy-to-use scripting language. Again, they have documentation, training, and bias. Yeah, but it's also exactly. But anyway, evolution always goes wrong way. I mean, if you look at the revolution, it always goes wrong way. Tell me once that the revolution went right way. It's crazy. What a crazy audience.
I just want to point out.
Yes.
Let's not like destroy MATLAB.
It's not bad. Exactly.
I don't know a biologist who actually uses MATLAB. There are a huge number of people interested in this that don't use MATLAB.
MATLAB sucks.
People aren't going to do it. So we set it up, and I sat on my phone, and I just clicked through, and in the comment section, I just felt like writing it, although I'm a pretty avid user.
So there's a
We
pros and
continue
cons.
to debate. Yeah, pretty.
But still, no, that's fantastic. But still, there are a lot of lessons we can learn from MATLAB. I mean, however, whether we like MATLAB or not, that's not for us to judge, but we can learn a lot from them because they're successful, right? So you should learn from people who succeeded, like Steve Jobs or, I mean, his dad, or Bill Gates maybe. So what would be this ideal, I'm not sure how up-to-date this is. What would be the ideal specification of the cell-based modeling platform? So I think I like Chase in the sense that it supports many modeling methods, and I think our platforms should not focus on just one method, okay? It should allow easy import of experimental images. Scripting language, well, just like MATLAB or Mathematica would be nowhere without it, I think scripting language is essential, at least for fast prototyping. There are not too many biologists or people who are wanting to do models who are experts in C++. And just as C++ evolves into a fantastic language, which unfortunately becomes harder and harder to grasp, C++ 11, C++ 14 bring lots of features, but it makes also it's harder to use. This can be a barrier. Open source, modular, extensible, of course, by all means. Easy to use, we talked about it. It should also allow linking to other scales, okay? Because sooner or later we have to parameterize our cell behaviors using subcellular networks or other type of subcellular simulations, okay? Of course, we need to have decent PDE solver suite there, okay? Because all of our models require those. Ideally, community-driven and supported, just like ImageJ. So they are a success story too. So Kevin Elisari did a fantastic job. And this would be ideal, I'm not sure how achievable this is, standardized model description. So I talked about root, I'm not going to talk too much. Again, this is a platform which all physicists use, high-energy physicists, particle physicists. Nobody even dares to develop their own physics simulation toolkit. It's all there. So let's see. So I think I'm probably going to wrap up my talk with this slide. So first of all, copying successful solution is not a bad idea. I mean, nobody's ashamed of. So what did Steve Jobs say about it? That good artists borrow, great artists steal. Yeah. Of course. I mean, there we are. So standards, yes, but be careful here. I think focusing on tools and avoid, so if we can agree on core technology to avoid effort duplication and develop this technology that is a platform, I think we'll have a stronger case in funding agencies and in the perception of us as a community. We have to focus on usability. No more cryptic software. It can be super powerful. It doesn't matter. If it's not easy to use, nobody will use it. That's a brutal truth. And we find this each time we teach workshops. And you'll be amazed that people, smart people, who come to workshops, sometimes we have to start teaching them how to find files on their computers, because they might not be aware about concept of directory structure, file system. That's a problem. But you teach them, and after day 2, they can build models. So you have to start with very low expectations. Don't assume that your audience knows C++. Don't assume that they know how a computer functions. Start with very simple concepts. And of course, just as you do that, you have to provide training works of documentation and try to engage developers to create a vibrant community. So I think I'll leave 5 minutes for any questions, and thank you for your attention.